Why identifying lipid peroxidation matter? Have you ever run the same experiment twice but got completely different outcomes? One invisible factor often explains everything: lipid peroxidation. Today we will show you how to identify lipid peroxidation in biological samples using fast and reliable approaches that researchers depend on. If oxidative stress has ever confused your data, this is the video you need. What is lipid peroxidation? The core idea you must know. Before identifying it, you need to know what lipid peroxidation actually is. It's a radical-driven oxidation of membrane lipids caused by reactive oxygen species. This reaction disrupts membrane structure and produces unstable oxidized lipids. Understanding this simple process makes every detection method clearer, because you're essentially tracking the chemical traces left by this oxidative cascade. Chemical probes: how to identify lipid peroxidation with MDA and 4-HNE. Aldehyde markers like MDA and 4-HNE are the most common indicators. Assays such as T-bars or ELISA quickly turn these molecules into measurable signals. If you want a straightforward answer to how to identify lipid peroxidation in biological samples, aldehyde detection is the classic, fast, and accessible method. Advanced tools: how to identify lipid peroxidation with LC-MS Lipidomics. For researchers needing precision, LC-MS Lipidomics provides molecule-level detail. It detects oxidized phospholipids and peroxylipid species with high accuracy. If your work involves lipid mechanisms or delivery systems, this is the most detailed approach to identifying which lipids were oxidized and to what extent. How to choose the right approach to identify lipid peroxidation. If you need fast screening, use assay-based aldehyde detection. If you need depth and specificity, use lipidomics. Matching your research goal to the right method makes identifying lipid peroxidation simple instead of overwhelming. Bulk sciences, lipid products, and lipid-based delivery support. If you need high-purity lipids, customized lipid synthesis, or support for lipid-based drug delivery systems, Bulk Sciences offers a full-service platform. From lipid standards to formulation development, our team can help accelerate any project involving lipid oxidation or advanced delivery technologies. Contact Bulk Sciences anytime for expert lipids and lipid-based delivery system development assistance.